here. I'm very grateful for the invitation of Florence and Donata. I think this is actually, I love the topic of this school. I think there is a very powerful combination of topics for doing nice advances in science and biosciences as well, and physical science and so on. Thank you so much. As Donata was saying, our path crossed by several indirect ways, like always happens in science, I think. So for those who are students here, you will live these things, that you keep meeting people that know people you know and things like that, and it's quite a nice thing. But this is the first time we meet in person, and I'm so glad. It's an honor for me to be here. So as Donata said, I'm coming from Spain, from the Basque country. There I work at the University of the Basque country, in one of the campus that is in the city of Vitoria-Gasteiz, which is the capital city. Okay, and I work for Ikerbasque, which is the Basque Foundation of Science in the Basque country. So first of all, I will start introducing the topic of a lab on a chip, which maybe you are very familiar with it, or maybe not. So I will start with a very short introduction on what is the concept of lab on a chip. Perhaps if you know this field, you have seen this picture many, many times. I don't really like the picture, but I think there is no better picture to show what lab on a chip is. So I keep using it. It's a very old picture, actually. So the lab on a chip technology refers to the miniaturization of laboratory processes. Or said in a different way, I want to integrate all the steps that you need in an analytical laboratory into a single device. For example, we are talking always about analytical microsystems to analyze fluids. So if you think of a blood analysis or soil analysis or any other type of analysis of fluids, many times you need to collect your sample, prepare your sample, deliver the sample to the sensor, and then get a signal. So all these different steps that normally you would do in a lab with different equipment, you want to actually integrate in a single device and make all that protocol as automatic as possible. So we want to miniaturize the whole lab into a single device. And this actually has shown through to give a lot of advantages to actually perform chemical analysis, biological analysis, actually opening the way to make new type of analysis as well, due to the properties of the microscale. Of course, it's characterized by the manipulation of tiny volumes of sample. Okay, so that's one also of the advantages. And then it involves, it's very important, in general, lab on a chip technology involves the integration of sensors into microfluidic networks. So with microfluidic networks, we only have a cartridge where we can put small amounts of liquids, but we want that cartridge to be functional and to do something. So that's why we need sensors, and we also need very often actuators to manipulate the flow inside the chips. So lab on a chip actually is an enabling technology which has application in many different fields. It enables to create new type of experiment, but also enables to actually acquire lots of data. And it has application in many different fields like biomedical diagnostics, monitoring environment, sport science, food quality. I put you here some biological research, not only for rapid diagnostics, and also we see more and more lab on a chip instruments that go to the space for actually carrying out new type of experiments. And This is an emerging field for the last 20, 25 years or something, 30 years. And I'm sure you are familiar with many examples that are already in the market, no? Like, for example, very old, kind of old technology is the pregnancy test, which is comprised of a kind of paper capillary microfluidics combined with gold nanoparticles for biomolecules for signal. And it's exactly the test that we used for COVID. We all used that for COVID. So that I always say that that's 30 years old technology, and now we are working in the next technology, right? So there are some challenges. Wearable devices, it's possible that somebody in this audience is wearing nowadays one of these glucose sensors commercialized by Roche to determine glucose levels to monitor diabetes. But also we start to work on sweat analysis with these wearable devices, and there are more and more. A very, very strong topic that is emerging for the last 10 years is the organ on a chip, which basically is trying to deliver platforms where we can mimic certain cell environments in more or less complex environments in order to create in vitro platforms where to test drugs. And this has it could have a lot of impact in pharmaceutical companies, reducing the cost of research for developing drugs, and also for example, the use of animals for experiments. Maybe many people here or not are familiar with digital PCR, but it's another example of the application of microfluidic technology to real life problems nowadays. So we established the microfluidic cluster in the University of the Basque Country in 2015, like Donata was saying, together with Fernando Benito. And there we built a very multidisciplinary team, because in order to do what I was saying, we need actually a multidisciplinary team. We need people who are expert in biology, biotechnology, chemistry, engineers, and I think even pharmacists are part of the team. So it's quite a multidisciplinary team, and we focus on two main topics. We focus on developing lab-on-a-chip technology with two main applications. One is rapid analysis, so devices to have a rapid analysis of a fluid. For example, I give you there an example with a test to detect abused drugs in drinks. So the idea here is to dip the stick in the drink and to get an answer if there is a drug or not in the drink, which you may know that is becoming a big problem nowadays. So that's an example of rapid analysis, right? And then the other topic we try to develop is platforms for cell analysis. Here, in order to do that, I will show you that you need, as I said, to develop fundamental components of microfluidics, and then we need to actually integrate all those components into useful microsystems. So here I show you a number of examples developed in the lab, where we are actually developing fundamental components. You see here manipulation of droplets with magnetic forces using ferrofluids. You see here valves, which are actuated optically. So those are polymeric materials that in the presence of light, they change the morphology, and you can open or close channels in a microfluidic network. Here, what you are seeing is a micro pump made of polymer, which actually drives the fluid through the channel. So this is important because it's an architecture for self-powered microfluidic devices. Normally, you want to minimize the amount of instrumentation you need around the microfluidic device in order to make it useful at the point of care. So these pumps were developed to make self-powered devices. And what you are seeing there, for example, is the separation of whole blood and plasma. So from whole blood, we can obtain the plasma directly. This would be equivalent, for example, to taking the sample in the hospital, going to the lab, centrifuge the blood, and separate the plasma. Right? You see an example of paper microfluidics, flow control on paper microfluidics by combining polymers and paper. Also, other type of gels and ionogels to create again valves inside the microfluidic devices. In this case, a three-dimensional microfluidic device. And here what you see is a chronosampling of a fluid by combining different sensing materials with a microfluidic structure. So these are examples developed in the lab, and they represent basic fundamental development of components. But we try to, because of the nature of what we do, our main objective actually, well, our main objective is everything, right? We do research, which is funded by research agencies, but we try also to do technology transfer. So we try to protect the components that we develop, and we have tried to start several, well, we didn't try to start several enterprises, but we support the formation of the company. So far, the only one that is working is the test for SAFE, which is this strip for the chemical detection of drug abuse drugs. But today I wanted to talk to you about that one other objective, which is to develop universal analytical platforms for study cell biology, which I think is more related to this. Basically, our objective is to create microsystems that can serve to a range of applications. So we want to develop the technology, but in a way that it can be easily used by non-experts in the field of microfluidics, because we want to develop things that are affordable, so that it can be used in mass. So it also must be something that can be mass produced, because in this way we can deploy and many people can use those instruments. In that way we can generate new data. No? And as we see, the more data we get, the more models we can create and the knowledge we can acquire. So it's very important for us to build platforms that other people can use and other people can adapt to their own assays and experiments. And also to make something that can be scalable and commercialized to actually be used a lot. So our approach to develop these analytical platforms is to develop knowledge in all these 4 different lines, and to combine them to create analytical integrated microsystems. So we work on microfluidic components, we work on functional materials to create sensors and also actuators, surface engineering, and integration of cells on chip. So I want to present today to you 3 different types of platforms we are working on, which are the SCADA platform, single cell addition array, the Cell Studio platform, and the cutoff, which is referring to quantum electron transfer, I tell you later, optical fibers. Okay, a key point on all these platforms is the use, or all this is enabled thanks to the microfabrication capabilities we have developed in the last 40 years or 90 or 50 years. And what I show in this slide is just to give you some examples to put your mind on those type of dimensions, as you know here very well. Nowadays, thanks to the microfabrication capabilities, we can assemble nanomaterials in a way that we can create microstructures, and actually we can use those microstructures or those capabilities for patterning surfaces to actually control addition of cells on surfaces, right? So in particular, we use a lot surface patterning for these objectives. And what I introduced in this slide is the concept of microcontact printing, which is also not new, has been developed 20 years ago. I'm not sure if you are all familiar with that, but it's a stamping technique to make patterns, chemical patterns on a substrate. So if we have a stamp, if we have a stamp and we ink the stamp with an ink, in this case blue, I can transfer the blue ink from the stamp to the substrate. That's what we do in the post office, or what we do. If my stamp contains microstructures, which I can create by photolithography and molding, and my ink is a protein solution, I can create protein patterns on a glass substrate. Proteins adhere very nicely to glass, as many of you know. And as you can see here, we can very nicely replicate any pattern we can create by photolithography. What you are seeing in the pictures is a glass slide functionalized with a pattern of fibrinogen, and the fibrinogen molecules has been stained with immunostained, with an antibody. So that's why you see the green. Where you see green, that is fibrinogen. In order to make this surface functional, we have to block the part that is black in the image. Actually, is blocked with a, for example, BSA protein, just to avoid interaction in those areas. If we take those substrate that has been patterned with proteins and we incubate with cells, cells actually will interact with whatever is in the substrate. They see the BSA, they don't like it, they don't adhere, but they see the fibrinogen and they have higher affinity for fibrinogen than for BSA, so they go directly to the fibrinogen. In this case, what you are seeing there is platelets adhering to the fibrinogen patterns, or fibroblasts adhering to fibronectin patterns. And what you see in here is that we have a good control on where the cells are adhering, and also on how many cells are interacting together. We can go from single cells to a number of cells together. So we can control cell-cell interaction. Not only that, but by introducing the protein that I want on the surface, I am also introducing another chemical cue in the system. If I change the protein, the cell behaves differently. That's a clear, for example, in platelets, if you add fibrinogen to the surface, platelets will spread. But if you capture your platelets with an antibody, they will remain like a ball, because there is some signaling going on. So you're introducing chemical cues through the surface. And because we want to make things which are scalable and affordable, and we want to mass produce these devices, so instead of using glass, we started to explore using plastics for doing these patterns. And I show you here several types of cells, patterns made on PMMA in this case, a plastic with several types of cells. And as you can see, the shape and the amount of cells that you put together is something you can choose. So this is a versatile tool. We can use as ink, we can use protein solutions, but also matrigel has been used, and we can select the substrate that we want. Glass or plastics, cell culture plates could be used. But okay, actually, I wanted to put attention to this picture. What you see in there is an array of single cells. Okay, by controlling the size of the pattern, I can control the number of cells in each area. So we decided to do arrays of single cells. To study single cells could be, but no. What we found is that by doing these arrays of single cells, we got a very easy way to quantify the number of cells that are adhering to a substrate, or that are captured in a substrate. And then, to prove that I'm right, I ask you to please count now how many platelets, in this case these are platelets over fibrinogen, are in this picture. And any of you will give me a different number. Instead, if I ask you to please tell me how many platelets are in there, taking into account that only one platelet fits in each dot, everybody will tell me the right number, or at least our error bars will be tiny. And this is exactly true. What you see here is the protein pattern, and what you see here is the bright field optical image of the plate. In this case, these are mesenchymal stem cells. Mesenchymal stem cells adhering to the pattern. So you can immediately relate the number of available dots and the number of occupied dots. So this, that is an analogical system, an analogical problem, now we made it digital, and we only need to quantify empty or occupied dots. 1, 0, 1, 0. So it's a very easy way and very precise way to quantify number of cells adhering to a substrate. So what we did was to integrate this idea into microfluidic platforms, and we do that in a very simple way. We have a substrate with the pattern of protein pattern, and then we use thin plastic layers that we can easily cut to create a fluidic gasket. And we create what we call the SCADA device, standing for single cell adhesion arrays. So what you see in this video is a cell suspension flowing over the pattern of fibronectin in this case. And the cells are mesenchymal stem cells. And you can see how the cells are adhering to the fibronectin dots, filling the array. And we can actually monitor this on real time, and we can build the adhesion curve. We can see in time, in a 10 minutes, 20 minutes time, we can monitor that adhesion curve. The interesting thing of this is that that curve will have a different type of shape or a different final array occupancy depending on the affinity of the cells for the substrate. So what you are seeing in this graph is what happened when I modify my pattern, and instead of having fibronectin, which the cells have a high affinity for it, for example, I put PEG or BSA. I can see how the cells cannot actually fill, they do not have the affinity to fill the whole array, and I can easily quantify that in a 20 minutes time. So this device is useful for at least determine different affinities of cells for matrices. What we are actually measuring is cell capture by the matrix, but eventually we could also start to measure to study cell adhesion effects. And we also applied this device to measure cytotoxic effects. So once you have the whole array is filled, if you expose that array to a toxic compound, what happens is that the cells detach. The cells detach. So the same way that I can quantify how they are adhering, I can also nicely quantify how they detach. The more toxic, the faster they die, the faster they empty the array. So this is a cartridge that people could use to study cell adhesion, cell affinity for different matrices, or cytotoxicity of different compounds. But keeping with the surface engineering, let's think if we can add some complexity. What I show you is a pattern. Before I show you a pattern of protein where all the single points contain actually the same protein, right? And I can do the type of measurements that I show you with the SCADA. But can we add complexity? Can we introduce something else in the surface? Because these proteins on the surface are very useful. They help me to direct cells wherever I want. So we wanted to do that, and for that we developed a new soft lithography technique, which we called printing and vacuum lithography, which combines microcontact printing with vacuum lithography. And vacuum lithography basically helps or enables to fill the gaps of a PDMS stamp between the areas that are in contact with the substrate. So you see here the PDMS stamp, the green ink can be transferred to the substrate by microcontact printing, as we explained before. And now I can fill the gaps with liquid and deposit other reagents in my surface. And this is how we actually build this image where you see two different proteins in the same substrate with a very nice resolution, because we do have the resolution of the photolithography. So the green is one protein, the red is another protein, and the gap between these two is 15 microns. So how it works, the printing and vacuum lithography. So in the video, what you are seeing is a liquid that is getting into the gaps of the stamp that contains the pillars. So the stamp contains the pillars, and this, what is here yellow, here is red. As you see, the liquid is filling the stamp. So what we do actually is to take the stamp already assembled to a glass slide and remove the air of the stamp. The stamp is made of PDMS and there is air inside the PDMS. When we bring all these things to atmospheric pressure, the PDMS tries to recover the air. So that creates a negative pressure inside of the channels, and that's why the liquid can go in, as you are seeing in the video. Okay, there is only inlet, there is no outlet. This is full of air, but because it has been in vacuum, the PDMS stamp is trying to recover the air, is taking all the air that is inside the network, and that's why the liquid is flowing. In this way, we can create this type of patterns where we combine microcontact printing with the delivery of other components by vacuum lithography. Very important is that these are two images of the same area of the substrate. So the two different patterns are in contact with each other, one after the other. So in this way, we can actually create very complex drawings in a substrate. We can actually, now it's versatile, and I don't know, sometimes I use this word universal, I think maybe it's too much, but I feel it is almost universal. You can draw anything in a paper, and basically nowadays there is the capability to translate that into a microstructure in a substrate, using, for example, protein solutions, or polymer solutions, or biomaterials, or whatever, anything that can flow into. So as you see here, we can create very complex structures. We can have different components. Here, what you are seeing is that we used different type of gold nanoparticles combined with the microcontact printing of fibronectin. So the gold nanoparticles, depending on their functionalization, interact with each other in a different way. And that's why we can create different structures. So we can control the addition of cells and combine with different type of metal nanoparticle structures, of gels or biomaterials or beads or whatever. You see here single cell array where each cell is surrounded by gold nanoparticles. Why we wanted to do this? Because gold nanoparticles, as you know, they have very interesting optical properties, and it can be used as sensors and eventually also as actuators. And you see here a collection of different cells placed in different environments with a microstructure of gold nanoparticles around the cell, or passing through the cell, etc. So it's a toolbox. But okay, we want to create a platform to monitor cells, eventually, hopefully, under controlled microenvironments. As you know, cell behavior is completely related to the microenvironment. The microenvironment modulates cell behavior, and the cell itself modulates the microenvironment. So the microenvironment is essential, right? At the same time, we want to create something that is simple enough to give us lots amount of data in a simple way. So we thought of exploring the possibilities of using this printing and vacuum lithography to have some type of substrates where we could obtain good data on cell behavior under controlled microenvironments. To do that, we divided the microenvironments in a very ignorant, simple way that I'm happy for you to actually criticize. No, like let's see how much sense it makes. We divided the microenvironment into the more fundamental parameters. We said, okay, microenvironment is contact between cells, cell-cell contact, is the interaction of cells with the extracellular matrix, and is the interaction of cells, well, with the topography of the extracellular matrix and also with the chemistry, right? So what we did was to have an array of adhesion zones for cells surrounded by beads. Beads that can actually act as a transporter of sensors, like antibodies, to capture molecules released by the cells, or actually could be used as a solid phase to expose chemicals to the cells. And that is what we did, and we called cell studio. So the idea is actually to just modify the bottom plate of well plates with areas where I can adhere cells in a very controlled way. And as I show you, those adhesion areas could be smaller or larger, so I can control cell-cell interaction. Then these adhesion zones where the cells are adhering is surrounded by microbeads that we introduced by the vacuum lithography. And these microbeads are microbeads, and they are polymeric beads that can actually be functionalized with molecules. Could be molecules that we want to interact with the cells, or it could be antibodies to capture, for example, proteins secreted by the cells. And it can be anything. It can be aptamers, or it can be gold nanoparticles, or it can be any other thing. The good thing is that we can create, because this is all by microfabrication, so we can have hundreds of these adhesion zones in a single substrate. So in a single way, we can actually measure hundreds of times the same events. And we can have a very nice statistical understanding of the cell population. And the other thing is that if we are able to capture the proteins released by these cells, actually we are measuring the secretion of just a very small amount of cells. So we could have cell behavior analysis, because we can look at by classical optical techniques. We can understand viability, proliferation, morphology, and secretion in a single substrate for the whole population of cells in one single well. So this is a zoom of what I was saying. So this would be the black area here is functionalized with any protein of choice. The cells are adhering to that protein, and then is surrounded by beads, like are in intimate contact with the cells. So I show you one set of data, and this is to prove that actually we can control those fundamental parameters of the cell microenvironment, and we can actually detect cell secretion. So in this case, mesenchymal stem cells were arrayed on the substrate, and the beads contained two things: antibodies to detect VEGF protein, vascular endothelial growth factor, and other beads were functionalized with the FGF factor. So the FGF2 induced was there because we wanted to induce higher proliferation. and the VEGF capture antibodies were there to detect VEGF secreted. What you can see in here is that in the absence or in the presence of the bits functionalized with FGF, the response of the cells is different. The proliferation is higher in the presence of the FGF, and also the amount of protein secreted by the cells that here you can see in green after immune staining is higher in the presence of FGF. So this demonstrates that, well, you can easily see proliferation by optical techniques. I didn't show it in here, but we can also see intracellular expression of characteristic proteins upon the exposure of FGF. And we can also detect the higher secretion of VEGF from the cells. So this demonstrates that there is a real interaction between the solid phase, the microbits that we put in there, and the cells. And then we also have data showing that the behavior is not exactly the same if there are 4 or 5 cells or actually only 2 cells. So by controlling those fundamental components of microenvironment, we hope we will be able to analyze cell behavior. But at least this is a multidimensional analysis of cell behavior with normal techniques just by modifying the substrate of the culture well plate. And to finalize, the other topic I wanted to talk about was ultrasensitive sensors made with optical fibers combined with gold nanoparticles. Because as I told you, it's for us very important to integrate, and this is one of the, I would say, is one of the most important points to develop in the future for all these organ-on-a-chip devices, is to be able to integrate sensors into the devices. Because otherwise, it's very hard to collect knowledge, no? You need to understand what is happening. So one of the sensors that we could actually introduce in these devices are optical fibers, because optical fibers can be very small. The ones, for example, we are using, they are common in telecommunications, but basically they are 100 or 200 micrometers diameter. So you could think of having many of these fibers plugged in a device, giving you the real-time data. So to introduce this topic, I need to explain to you something that was developed and published by the group of Luckey in Berkeley already in 2007. I think here many people is familiar with plasmonic nanosensors, referring to the optical properties of gold nanoparticles. And as you know, gold nanoparticles, they absorb light and they also scatter light, and they have a characteristic plasmon peak, which is known as localized surface plasmon resonance. So this is a typical image of gold nanoparticles on a dark field microscope. And the dark field microscope is able to actually shine light on a sample, avoiding that the light of the source is arriving to the detector. So what you can measure, what you actually measure in the dark field microscope is only the scattered light. If you shine light on gold nanoparticles, part of the light is absorbed and part of the light is scattered. So that's what you observe. And that's why we observed in the microscopic image different colors from the nanostructures. Because depending on the size of the nanoparticle, you get actually different scattered light with different wavelengths. That's normal plasmonics and scattered light. This system is around 200,000 to 300,000 euros. And what Luckey discovered was that if there is a protein on top or a molecule on top of a gold nanoparticle, and that molecule absorbs light, has a UV spectrum, absorbs light in the same wavelength that the gold nanoparticle scatters light, they actually show that there is a energy transfer between the particle and the molecule. Because when they looked at the spectra of those nanoparticles that had the protein on top, they actually saw the UVV spectra of the protein inside the spectra of the nanoparticle. Okay, and this was, to me, it was very exciting because it's like they were working as nano antennas, and they actually lowered the limit of detection of the UVV spectroscopy a lot. And then they started the field where they could detect very small amounts of molecules because of this interaction and energy transfer with optical gold nanoparticles. Okay, and they did all this with a dark field microscope. Okay, and they were able to actually measure the cytochrome C production in cells that were dying, so intracellular. So I was actually working with Luke Lee, and then all these ideas were in my mind, like let's try to combine everything. Let's have nanoparticles on the surface so that we can go one time after the other. Let's try to detect cell secretion with these nanoparticles, because at the end, this is actually a label-free technique. You are seeing the spectra of the molecule, you look. So you don't need to stain, and you don't need, it's like almost SPR, but actually you get the spectra of the molecule. No? It's like similar to SERS, but it's UVVs, and you use only white light. So for a long time, I was trying to actually to reproduce this, but with this type of patterns. I said, okay, we can put cells on a surface, we can surround the cells with gold nanoparticles, the cell will secrete proteins, and we will detect them in a label-free manner. No way, I couldn't. I keep trying, I keep trying. But then a collaborator who was actually working on photonics and optical fibers, we were discussing, and actually we were trying to build a system to do this, but we couldn't. And suddenly he told me, but I want to do something with polymers and optical fibers. And I say, optical fibers are made of what? Of glass. Oh, glass. We know how to functionalize glass. So the idea was, can we actually functionalize the glass fiber tip with gold nanoparticles so that we can measure the optical properties of gold nanoparticles? And that's what we did. So the idea was, if we are able to actually coat the tip of glass optical fibers with gold nanoparticles, perhaps we can see the plasmon peak of the gold nanoparticles and then study their properties. The system now was comprised only of a white light source, it's an LED, the optical fiber, and the spectrophotometer. And this whole thing, just bought for a lab, is around 2,000 euros instead of the 200,000 euros of the market. And it has a coupler here. So basically, as you see, it's a very simple system. We are using multi-mode optical fibers. And this is the type of data we can get. We can see very nicely that the amount of gold nanoparticles on the surface is increased the intensity of the peak. It makes sense. Okay, and we can relate actually the intensity of the peak with the amount of gold nanoparticles in the optical fiber. Here you see the SEM pictures where each dot is gold nanoparticles. Okay, so the next thing is like, okay, we can have the spectra. In this case, they were like 40 nanometers gold nanoparticles, and we were getting very nicely the plasmon peak of the gold nanoparticles. If we add here, I didn't add this, but we did it already. If we add different type of gold nanoparticles, we actually see all the peaks of the different type of particles. For example, if we add stars that has a plasmon peak around 600, or we can actually see the two peaks. So this is a very nice also way for multiplexing. So the next step was, the next and final for the moment was, okay, let's try to observe this energy transfer between gold nanoparticles and molecules through the fibers. To do that, we had to modify, to chemically functionalize gold nanoparticles with different type of molecules to be able to complex, for example, metal ions. So we functionalize gold nanoparticles with a kind of butylamine to complex copper. And you may know that the copper complexes, they have a UV-Vis absorption spectra around 550. So what we saw when in the presence of copper is that the typical plasmonic peak of the gold nanoparticles was actually decreasing and was shifted to the blue, indicating the presence of the copper. If we wash the fibers and we remove the copper from the gold nanoparticles, we actually recover the spectra typical of the gold nanoparticles. The very interesting thing about this is that our sensitivity was 3 orders of magnitude higher than the sensitivity obtained with the dark field microscope. And to me, this makes sense because when you have the microscope, the light has to go through the slide, the objective, and all the optical path. So you lose efficiency. Instead, when you have the optical fiber, the light goes directly from the source to the tip and go back to the spectrophotometer. So you don't have so many losses. And that's why we think we could increase the sensitivity of the system. Finally, we did another experiment. Also, it's a replica of the experiments made by the dark field microscope by Lourdes, where we functionalized with a molecule that was able to interact with cytochrome C. And upon the interaction of the cytochrome C with the gold nanoparticle, we could very nicely see the spectra of the cytochrome C through the spectra of the gold nanoparticle. We could also detect this cytochrome C in plasma samples. And I think this is interesting because we have a very clear peak of the cytochrome C. And people ask me, how is possible that you get that if there are so many other proteins in plasma? And this is correct, but most probably only the cytochrome C is the one that overlaps the UV-Vis spectrum with the scattering spectra of the gold nanoparticles. So that's if we open the range of the gold nanoparticle spectra, we will be able to see more proteins. So more proteins interact with the particles, but we only see the cytochrome C. So this is the end. I wanted to talk to you about these 3 platforms. To remind you, our goal is to have cartridges, devices that can be easily used by other people and can be adapted to the assays of other people. Thank you very much. You can take a look at our webpage, and thank you very much for your attention.